I'm very happy to be here. Hope to see you guys bright-eyed, bushy-tailed, ready to talk about emulsification at 8:30 a.m. My name is Camden Cutright. I'm here with my colleague Claire. We're representing Micropore Technologies. Micropore is a chemical engineering company, and we help people develop better processes for encapsulation. Now, the first question we always ask anybody when they come to us is, why are you encapsulating? I mean, if your active works all on its own, it's shelf stable, can get where it's going and do what it needs to do, just take your 4-leaf clover, your winning lottery ticket, and whistle on down the street. But very often, what we're looking for is some kind of additional functionality. This could be a control release, whether it's temporal or environmental. You could be looking to have a specific tissue that you're looking to target, protection from oxidation or preserving reactivity, different kinds of flowability properties. There's all kinds of reasons that people may want to encapsulate. Now, in addition to some of the technical reasons you might want to encapsulate, you also need to take into account what will make my product marketable. So we call these the non-negotiables. And it's the marriage of why am I encapsulating, what does my customer want, and what do the regulators allow me to do. This will vary product to product and industry to industry. But if you look at an agrochemical product, for example, you might want to be preserving your reactivity over a long period of time. You could be manufacturing in the winter in Ohio, shipping down to a farm in Georgia where your product sits in a barn all the way through July before it's shot through a nozzle onto a crop field. And it has to survive that entire process. But remember, we're selling to farmers here. We can't have the product itself destroying crops. And the farmers can only sell their crops for so much money. So if it costs you more to manufacture your microcapsules than they can make from selling their crops, you're going to have an imaginary customer. And finally, you may want to take into account the kind of materials that you're using to encapsulate. How does your local geography view things like microplastics? It can be tempting as you're doing this work to sort of dine from the microencapsulation possibilities buffet, start adding more and more to this list of non-negotiables. But what this really ends up doing is boxing you out of any kind of product that you'll ever discover. So if you want to not have an imaginary product with imaginary customers, be as ruthless as you can with determining the barriers of your design sandbox. Now, you've got plenty of leeway here most of the time to set a few boundaries in place and then optimize for a couple of different parameters at the same time. You just have to understand that this is going to result in some trade-offs. So let's talk through a morphology example here. Let's say you have a wonderful spray drying process for making a matrix particle, but you come up with an application that might need a coating. Is it possible? Of course. Talk to the guys at Inc.at or Experian, and they can give you lots of examples of how they onboard a new process, maybe a fluid bed coater, to add a coating to your matrix particle. Now, what's it going to do? It's going to cost a little more. There's some obvious costs here, things like the capital equipment, the extra shell material, but there's some hidden ones as well. It's the idea that, well, now we have a new unit operation. Do we have the expertise to bring that on? How much time are we willing to spend re-optimizing? And pro tip number one for this talk is, whenever you're going to do something like this, make sure you find the right partner. Because spending a little bit of money to save the time and energy, finding somebody that knows which my nozzle will be on top or on the bottom or on the side, how quickly can you coat, how many layers can you coat, that's going to save you way more than onboarding yourself. Let's look at another example, maybe particle size. Who here has heard the question, can we make it smaller? Pretty much every system has the same answer. Absolutely. Of course. Yeah. Maybe. And no. And as somebody that's worked in sales before, I can translate maybe for you. It means one time for one customer, one formulation that's not yours kind of worked, but it was hard. So, let's say you've got either a matrix particle or a core shell system, somewhere around 100 micron range, could vary technology to technology. You're going to get into that maybe range. So where emulsification comes in, where we see quite a bit of customers joining us, is, can you make the particle smaller? Yeah, sure. It's relatively easy to do with emulsification. What's the trade-off? Well, it's the carrier fluid. So if you're using a spray dryer, the carrier fluid disappears during the formation process. With something like a dripping technology, usually the particles are big enough that they'll settle at the bottom, and you can pour off a large volume of your carrier fluid. With emulsification, it's a feature, not a bug, that you end up with carrier fluid, right? Because you're trying to make small particles, they're small enough, they're colloidally stable, they stay suspended during the emulsification process. If they don't, that's a bad thing, right? You're going to get phase separation. So you just have to understand that the trade-off for the smaller particles is now I have to deal with this extra carrier fluid. And there's ways to do that. Now I've talked a lot so far about processes, and we'll get back to process in a moment, but understand that raw materials are just as big a component of building out your formulation as the process itself. Who here can tell me what these guys have in common? There are at least 2 correct answers. 1, they're both delicious. Number 2, they are of course both gelatin, or at least precursors to it. And it's probably not that hard to intuit that farm to farm, season to season, certainly species to species, the quality, the viscosity, the bloom strength, the gel strength, even the melting temperature of that gelatin is going to vary a little bit. And this is the case regardless of what raw materials you're using. You can talk about alginate, agitation, hyacinth. No matter what you're doing, you're going to end up dealing with resources that are coming from different parts of the world. You could have one government that decides to invade their neighbor, or maybe impose worldwide tariffs. And you're going to have to understand how to deal with those perturbations in quality and cost over time. You can't escape this, by the way, using synthetic materials. People are coming around to the idea that it may not be so great to coat the world in thin films of microplastics. So just be ready, regardless of what process you decide, what products you decide, to play with those trade-off relationships inside your design space. Now I know we want to move towards emulsification. I promised that at the beginning. So this is my egregiously oversimplified cheat sheet of where you might use an emulsification process. Generally speaking, people like physical encapsulation when they can get away with it. Aside from maybe pharmaceuticals, the encapsulation market is largely a low-margin, high-volume market. There are some places where that's not the case, but in general, because of the carrier fluid that comes with emulsification and other chemical encapsulation processes, it's cheaper operationally to use a physical method when you can. When might not you be able to? Well, if your core solution is volatile, you may have trouble spray drying it. If you're trying to make particles smaller, you may need a chemical encapsulation process. If you're looking for a complex particle morphology, something entrapped within something else, then you're probably going to need a more precise process. So those are some of the reasons that we see people using chemical encapsulation, but it really varies product to product, and it's very important to find trusted partners, whoever that is for you, to talk through and see what materials and what processes are going to work best for me. It is possible as well to do a bit of nanoparticle work with different chemical processes as well, but that's probably outside the scope of what we're talking about here. If you've got any nanoparticle questions, come talk to me later. Now, finally, on to emulsification. Emulsification has been around for a really long time. I've seen publications and patents coming out from the 1950s, probably as earlier than that. And emulsifiers originally were done with homogenization, and they're still done with homogenization today. There are tons of different ways that you can homogenize, but the general principle for all of these is roughly the same. We're going to take 2 immiscible phases, combine them together with energy, and that's going to separate the lower volume phase generally into the higher volume phase. Now, if you just wait a couple of minutes with nothing else, they'll phase separate again. But if you add some stabilizer, you can preserve that metastable state for some time. What is this good for? Well, we can make capsules of a fairly wide size range, and we can tune that size range with how much energy we input. So if you want to make a 30 micron particle one day and a 10 micron particle the next, you just have to spin a bit faster or input a little bit more energy. The equipment is fairly accessible. Most tool manufacturers already have this in-house, and if you want to bring a benchtop unit in, you can do that very easily. This has worked for a long time. I mentioned since the 50s. You can see applications in coatings, agrochemicals, pharmaceuticals, all over the place. And there is, generally speaking, a pathway to scale. Like I said, most tool manufacturers are going to understand how to take something that starts off at the benchtop and scale it up to pilot and commercial production. Okay, homogenizers sound great. Are there any problems? Well, not necessarily, depending on your application, but in other cases, yes. So what happens inside a homogenizer is that you input energy to break up the droplets. That's great. The closer you are to the source of that energy, the smaller your droplets are going to be. The further away you are from the source of energy, the bigger the droplets are going to be. And that inherently creates a relatively wide particle size distribution inside the device. And you can't really escape this by switching from one kind of homogenizer to the other. In some cases, your product quality is going to end up being tied relatively tightly to that particle size. You're going to have what we like to call the Goldilocks zone, where some of your particles are the right size, others are too small, others are too big. Now, in some cases, you have to filter those out, which is an extra processing step. In some cases, they're just waste. It can also be somewhat difficult to maintain your batch-to-batch reproducibility of the homogenizer. I don't know how many of you have baked a cake before. You set it in, and you wait 30 minutes, you check it, you put your fork or your toothpick in, and there's still batter. Darn, okay, put it back into the oven. 5 minutes later, it's basically lyophilized. It's just a big, dry rice powder cake. Oh my gosh, I messed it up. The same thing can happen here if you don't have great control over your homogenization process. Because it's batch, and you can't see exactly what's going on, it's very easy to under or overshoot your specification. Finally, if you look at the pathway to scale, which originally was a pro, it's also a bit of a con. And the reason is these two unit operations don't necessarily look exactly the same. And when you want to go from stage A to stage B to stage C, scaling up your production tenfold and hundredfold, it's non-trivial to make sure that those unit operations are working. So, is there a way that we could operate continuously, that we could get precision control over the particles? Yeah, sure. It's called microfluidics. Right? This was invented in the 70s, the 80s. People use very small channels, very precision flows in order to get extremely uniform particles. I'm not going to go over this in extreme detail. Our friend Anshan gave a great overview of how microfluidics works yesterday. But you get continuous flow, you can work with a variety of materials, control particle morphology to an incredibly high degree. The downside is physics tells us that each of these channels can really only flow so fast. If you want to keep increasing the velocity within a single channel, the pressure is going to rocket up. And even with some of the modern devices that have parallelized channels on chips and multiple chips stacked inside the same operational unit, you're only looking at maybe kilograms per day of production. So, where does Micropore come along in all this? We developed in 2017 a continuous version of what's called membrane emulsification. And this is essentially a long tube that we have drilled multiple channels in, anywhere from hundreds to thousands to even millions of microfluidic channels into a single stainless steel housing. And this allows us to sort of bridge the gap between the precision of microfluidics and the scale of conventional homogenization. The flow, your dispersed phase, your coil phase, comes in through our device, flows through the channels, and meets with continuously flowing carrier fluid. The speed of that carrier fluid and the size of the holes determines exactly what kind of particles you form. And when you compare the results of membrane emulsification to something like conventional homogenization, you can see that we're getting a much tighter size distribution. And like I said, depending on the configuration of the device and its operation, you can still tune that D50 to different locations while maintaining a tight size distribution. The question is, of course, does it actually scale? And it has a relatively simple scaling principle. When you want to go from benchtop, making 100 mil a minute, you need very few holes, very little porous area, slow moving pumps. When you want to scale up, you simply increase that porous area and use higher capacity pumps. It's as easy as that. If we can go from operating maybe 100 milliliters a minute on the bench to over a ton per hour at commercial scale. If we look, for example, at how this actually works, I haven't given you any data yet. You need some reasons to believe me. Let's take a couple of academic papers that have come out around things like coacervation, cell encapsulation, and sustained release. We'll take coacervation first. This is a paper looking at curcumin encapsulation within a coacervator. The authors here, I don't think, did a particularly exceptional job at using the homogenizer to encapsulate curcumin. They were only able to get about 40 to 60% loading. I think a more skilled operator probably could have done a little bit better. But even still, if you're looking at what happens to that capsule after it's brought to the body, how is the bioavailability going to be affected depending on the particle size, the shell thickness, the consistency. If it turns out to be the case that you're giving somebody a dose that's 10 grams, but because some of the particles never rupture and some rupture too early, they're really only getting 5. Imagine instead if everything was in the right size distribution, it was all delivered to the small intestine just at the right time and under the right conditions. And that's what we can do with membrane emulsification. We can make sure that every core is the same size, every shell is the same thickness, everything has the same strength. We can even find ways to make this process continuous by taking the coacervation process that begins with emulsification, but then goes downstream into temperature and pH control through a long plug flow reactor in order to make a fully continuous operational process. This is massively beneficial, right? Because you can check how things are going during the entire manufacturing process. You don't have to wait hours to see, did my coacervate work? Have I started agglomerating? If something's wrong, just divert the stream for a little bit, make an operational change, and keep going. Let's take another example here where the students were encapsulating yeast cells within a gelatin matrix. This was done both on a rotor-stator homogenizer, operating at roughly 6,000 RPM, and through our membrane emulsification device. Both targeted the exact same D50, but it turns out that the rotor-stator broke up some of the particles, we lost cells into the oil by comparison, and also the shear force that was imparted during the particle formation process ended up killing some of the cells. And at the end of the day, the membrane emulsification process meant that on a kilogram of gelatin to kilogram of gelatin basis, you had 4 times the total number of viable yeast cells. Now, if you're thinking about this as how much processing do I have to do, how much carrier fluid do I have to remove, this means that you're getting essentially 4 times the efficiency. And this doesn't even take into account the size distribution. Size distributions for this application didn't matter. And finally, let's look at an example of injectables. Something like the controlled release of a small molecule from an algae polymer matrix. The idea here is that you load your microsphere, and over time, the biodegradable polymer falls apart and releases your drug. Ideally, what would happen is you'd have some timeline over which you wanted this drug administered, You would want the patient to essentially immediately jump up to the therapeutic dose, not shoot above it, because anything above the therapeutic dose is just wasteful, and then maintain that dosage over time until at some point the particles are gone. This is actual customer data. I've removed the X and Y axis for their anonymity, but they produced their samples on their own homogenizer, and they asked us to make samples for them on our membrane emulsification devices. And what you see in green is the homogenizer results. So what happens here? Well, we start with a wide size distribution of particles. It takes a little bit of time for the drug to start eluting, but once it does, all of the particles are releasing components. Over time, the smaller reservoirs deplete and disappear, the bigger reservoirs continue releasing, until eventually you run out of drug altogether. For the membrane emulsification process, because all the particles start out the same size, you don't see this deteriorating release profile over time, not until essentially your process is designed to stop. I would hope at this point that membrane emulsification sounds a little bit interesting, at least to some of you that are doing chemical encapsulation processes. I do see some eyebrows raised here and there. So let me go ahead and give you the drawbacks as well. No process is perfect. Membrane emulsification has this wonderful size distribution control. We do that by imparting energy in a very controlled way. At some point, we can't continue to do that. We can't continue to put enough energy into the system. And so we have a minimum size, roughly 10 to 15 microns, or below that, if we're going to make our droplets, the size distribution is going to start to increase again. Number two, the pores that we're using, they're more like long tunnels than tiny holes. And what that means is if you're doing any kind of suspension-based processing or solids-based processing, it's very easy to turn this device into an expensive dead-end filter, right? So you have to be very careful with the shear thinning behavior, your disperse phase, and any solids that are present there. And finally, just like any other emulsification process, you're going to have to find a way to deal with carrier fluid. So with that, I hope I've given you an overview of microencapsulation via membrane emulsification. I've learned a little bit of something, and I'd be happy to answer any questions that you have.